I'm Francesca Wormsley, I'm from Vinidea. So we're supporting both the University of Milan and Stellenbosch University in the co-organization of this online event, and it's an honor for us. The person today who will be, in fact, talking through the whole event and presenting everyone to you will be Ileana Vigentini, who is the one of the principal investigators in this project and the coordinator of the project representing Milan. So I don't know, Ileana, if you'd like to turn your camera on in the meantime and just let everybody see you as well.
Thank you for the introduction.
There we go.
Maybe I can start sharing my screen.
Absolutely.
Okay, thank you. Okay,
Perfect.
you see my screen? Okay.
Absolutely.
Okay.
The floor is yours.
Thank you. So good afternoon, everyone. It's a great pleasure for me to welcome you to the final event of the BiSus4Wine project. It stands for Biocontrol Agents for a Sustainable Grape and Wine Production. And this project was developed within the framework of the Italy-South Africa Joint Research Program. In particular, this program aims to strengthening scientific cooperation between Italy and South Africa. We presented our project proposal in 2022, and it started in 2023, in early 2023. And these type of cooperation projects are organizing different funding schemes. But anyway, these schemes are implemented under a co-funding model between the two countries. So, for example, we have two schemes in this case. One is a collaborative research project scheme, and this means that indeed it includes clearly general and specific objectives and tasks, and indeed the research is co-funded by one party in at least in Italy and one at least in South Africa. And the other scheme is a research exchange program, and this is anyway co-funded, but it covers just the mobility cost. In our case, we applied for the first scheme, so a collaborative research project, and the project in this case, the scheme in this case, supports 3 years, a 3 years program, and it has different focuses in several strategic areas, agricultural biotechnologies or environmental biotechnologies or in general health. And our project was based on agricultural biotechnologies. And for example, here we decided to apply for our skills, of course, in the applied microbiology and in an approach that use and exploit microorganisms for plant and in some cases also on soil health. So our goal was within the BiSus4Wine has been to develop biological alternatives to fungicides, and this through the selection of and the characterization of specific microorganisms antagonistic with an antagonistic character against the proliferation of pathogens. Evodia, in this case, will show you more detail about the project, but in this case, I mean, the results lay the foundation for future biotechnological application, the development, of course, of bioprotective tools as alternatives to fungicides, and hopefully for future and potential patentable outcomes. So today, I would like to celebrate, of course, our collaboration, our 3 years of collaboration, research, and also the building up of a consortium in an international community focused on the development of sustainable agriculture, in this specific case, viticulture. So I warmly thank all the researchers involved in the Italian and South Africa teams, and of course the funding institutions, agencies, and all of you to be here today, because I mean, this means that there is a commitment, a strong commitment to a sector that is vital for our two countries. But starting from the Italian group, I will present, our team in Milan, at the University of Milan, with myself as the principal investigator, Daniela Fracassetti, Alessandra Di Canito, Tamar Magrazze, Elena Palencia Mulero, and Simona Pizzi, all collaborating in the preparation of microbial collection, the selection and characterization of interesting and attractive biological control agents. And we studied, for example, the interaction of BCAs with plants and pathogens, and also we focused our attention in wine fermentation. Also, again, from the Italian side, the University of Perugia with Professor Cardinali, Laura Corte, Deborah Casagrande, Angela Conti, and Brilli. They have skin in biodiversity analysis, BCA characterization, and wine fermentation. And while for the South Africa side, of course, the second PI of this project, Evodia Setati, Professor Bauer, Florian, Hevelyn Maluleke, and Christine Kruger, for the microbial collection, again, BCA selection and characterization, the study of microbial interaction and wine fermentation, and the University of Cape Town with Arina Hitzeroth, Malika Abrahams, and Axel Collins. Of course, they also were focused on microbial collection, the assessment of biodiversity, and again, the isolation of attractive BCAs. And finally, again, for South Africa, the Agricultural Research Council with Heinrich du Plessis and Justin Hoff, and all their team in the microbial collection, the assessment of biodiversity, and again, the selection of interesting and potential BCA candidates. So I would like, to conclude by expressing, of course, my gratitude to all the consortium partners and also to the funding agencies that all, of course, made this project possible in these 3 years. But it is now my honor to introduce Dr. Anna Quattrone, from the Italian Ministry of Foreign Affairs and International Cooperation, who is responsible for the Italy and South Africa Scientific Cooperation Program. I must add, of course, that the support given by the ministry has been essential for making these 3-year partnerships possible. So thank you very much. I interrupt my sharing my video, and I would like to give the floor to you, Anna. Hello, can you hear me?
I don't seem to see Anna in the panelists at the moment. She might have had an issue with her connection.
Okay.
Could be.
not sure if we have the second representative.
Sorry, we have always some problems of connection.
Okay. Hello, Anna. Thank you.
Hello, good evening. Good evening to everyone, and thank you very much for the invitation and for being part of this important event. On behalf of the Italian Embassy, first of all, I would like to congratulate Professor Vigentini and all project partners for the successful outcomes of BiSus4Wine. That is one of the 6 projects of major relevance funded under the Italy-South Africa Scientific and Technological Cooperation Programme. This project represents an excellent example of effective bilateral collaboration between the University of Milan and the Stellenbosch University, coordinated on the South African side by Professor Evodia Setati, with key contributions from other partners, and in particular the Agricultural Research Council in Freetech and the University of the Western Cape. BiSus4Wine addresses a major challenge for modern viticulture, reducing the use of chemical fungicides through sustainable biological control strategies. During the first phase of the project, 19 antagonistic yeast strains were identified as effective against Botrytis Cinerea, including strains such as Arabasidium pullulans, able to strongly reduce fungal growth, and species producing antimicrobial volatile compounds capable of completely inhibiting pathogen development under experimental conditions. This project really highlights how scientific cooperation can generate concrete impact by promoting more sustainable viticulture practices, supporting environmental protection, and contributing to the quality and competitiveness of wine production. From our perspective, the Embassy's perspective, BiSus4Wine is a clear example of science diplomacy in action, because it connects institutions, laboratories, and young researchers. It connects people and countries and innovative projects from both countries, building long-term partnerships based on excellence, sustainability, and shared innovation that are essential goals of our countries and of the era in which we are living. These results confirm the strength of the Italy-South Africa cooperation framework and its capacity to foster projects with real scientific value and practical relevance. So, thank you to all partners for this outstanding collaborative effort, and we really look forward to future joint initiatives building on this success. Thank you so much.
Thank you, Anna. So, unfortunately, Ileana, Dr. Nukutula Mchunu won't be able to join us.
I'm here, Francesca.
Oh, there we go. Sorry.
No, it's okay. It's okay.
Thank you very much. I didn't
No,
see you in
it's
the
good.
panelists there. Welcome to join us
Yeah.
then. I'll leave you the floor.
Yeah, I don't know. I see, because Evodia had shared her link. I think they had forgotten me, and then I got stuck in somewhere else.
It's absolutely fine.
Again,
Thank you.
good afternoon, everyone. Apologies for making everyone wait and making Evodia very anxious for not pitching up. My name is Nokutula Mchunu. I'm from the NRF, National Research Foundation, and of course, it is our partner. We implement all the bilateral cooperation by our Department of Science, Technology, and Innovation, and Italy is one of our priority partners. So for me, this meeting actually is a pleasure, because although now I call myself a paper pusher, it is microbiology is my first love, is my expertise. I have a PhD in microbial genomics, and at one point, I must declare in this meeting, Evodia, and I serve in the executive committee of a regional branch of South Africa for a microbial society for South Africa. So we come a long way while I was still in academia. So it's a real pleasure for me to be part of this meeting. But yeah, on the NRF, I think. Our cooperation with Italy is one we really appreciate it, and of course with Prof from University of Milan, and of course Evodia leading these two projects. I think my counterpart from the embassy has already mentioned the importance of this project in both growing the viticulture and making sure that we maintain environmental sustainability through using natural means in wine production. As you know, Stellenbosch and wine industry in South Africa is probably outside of Europe, is one of the most important and biggest. So we really this project is really important. But I will move over a little bit away from science and come to the meeting that we have here, which I think is important. So I just stepped out from what we call that our science ministry is hosting here at the CSR, the World Conference on Science Journalism, where we are talking about communicating science to our stakeholders, which is really important. And what we are doing here, this is important because, of course, Prof. Vigentini and Prof. Setati could have closed the project and disappeared and given us a report. But this is important to call the stakeholders and communicate the science, because this is the only way we could move forward in the cooperation, because we do science for the society, not science for only publication and communicating among peers. So I really do applaud and am happy seeing the stakeholder meetings happening. And also, I would really like, even as here we have young researchers, to really think about how do we communicate our science to the society or to the stakeholder that we make the project or the outcomes for, so that they can think about it. So it becomes a little bit also us thinking, how do we communicate our findings in the way that they will accept or can understand. So I do really appreciate it. And of course, our ministry now has been really big in thinking about impact, not just impact, academic impact, but the whole societal impact, and how we can make sure that the science that we do translate into that. So for me, I will keep this short, and hopefully soon with our partners in Italy, we will be renewing our cooperation, and we will be probably opening up new calls to follow up after this one. I think I will end there, and thank you for indulging me, and thank you, Evodia, for making sure that I do arrive here. It's really a pleasure to be part of this community, and of course, personally, it's a great pleasure. But also for the NRF, we really appreciate the invite. Thank you.
Thank you very much. And also for supporting this project. I mean, from both sides. I mean, from Italy, the Italian representative, and also the South African wine, because I mean, it is a strategic vision, and this means that it's paid attention to the scientific context, but also to the societal impacts, as you told us. So maybe for the new participants joining the meeting, Francesca, would you like to?
Yes, as a few
explain
more people
better?
have joined us, just a quick reminder before carrying on. During the 12 presentations, if there is time, at the end of each one, we will dedicate the questions that we receive to the speakers. But if not, if you can just write your questions in the Q&A section, specifying the name of the speaker you'd like to ask the question to. We will collect them all and dedicate that to the end of the session. So thank you, Ileana.
Thank you, Francesca. So it's now the time for Evodia Setati. She is the principal investigator for South Africa, and she will present us an overview of the project, its rationale, scientific objectives, and also the work plan developed by all the partner groups. Thank you. Thank you, Evodia.
Right, thanks, Ileana. Good afternoon, everyone. Yes, so as you heard from Ileana, I was tasked with just giving an overview of what this project was about, and I'll try not to repeat some of the information that has already been shared. Right, so I just need to move this. Go. Right. So I think most of you might wonder why this project. So as you've heard both from our funders that this is an important industry. I think most importantly for us is to note that the industry is important both in Italy and in South Africa. And if we consider, for instance, the statistics from the International Organization of Wine and Wine, we see that with a total vineyard surface area of 7.1 million hectares, Italy accounts for about 728 kilohectares, while South Africa accounted for about 120 kilohectares in the year 2024, which shows you how big the production is, especially for Italy being the third biggest country in terms of production and surface area. And in both countries, grape and wine production contributes quite considerably to the GDP, so the gross domestic product. And as you can see, for Italy, wine production or the wine industry contributes about 1.1% to the GDP, while in South Africa, it contributes about 0.9% to the GDP. Now, it's important to highlight that the production of good quality grapes and wine is not that simple. In fact, it's constantly challenged by plant pathogens that can lead to serious crop loss. So how do we address this? So if we look at the type of pathogens, one of the important ones is Botrytis Cinerea. And Botrytis Cinerea, for those who do not work in the wine industry or with fruit, it is a highly important filamentous fungus that infects different fruit crops, both pre and post harvest. And in grape and wine production, it's often referred to as gray mold or Botrytis bunch rot. And this is a pathogen that's important in all wine producing areas, irrespective of whether you're in the Northern Hemisphere or in the Southern Hemisphere. Generally, the spread of the disease, or even the beginning of the disease or the prevention of the disease, is managed through a combination of strategies. This can be through the removal of diseased parts during winter pruning to avoid an overload of spores within the vineyard ecosystem. And during different phenological stages, there's also application of chemical fungicides, usually from the beginning of flowering throughout the grape development and grape ripening, until closer to harvest. And in addition to this can be through canopy management, where leaf removal in the bunch zone just helps to increase air circulation or airflow around the bunches to limit humidity within the bunch zones. So while the use of chemical fungicides has been sort of the cornerstone of grape and wine production, it is not without limitations. So often we see that with a fungus like Botrytis Cinerea, the fungus tends to build mutations that allow it to have resistance to a variety of fungicides. And also the use of fungicides is becoming more and more a problem across all agricultural sectors due to the accumulation of fungicides in the soil, and therefore increasing risk for runoff into groundwater. Additionally, the fungicides have been shown, or some fungicides have been shown, to result in residues that remain on the grapes and therefore are transferred into wine. Most of these fungicides, however, have a 30-day holding period, in the sense that while they can be applied from flowering, you cannot apply them around 30 days within the 30-day period before harvest. And so with the challenges of climate change, where you have rain and heat resulting in high humidity closer to harvest, it becomes a problem to use these fungicides to prevent or even to control the disease. In addition to these challenges, I think we can all agree that there has been mounting pressures, either linked to legislation or just to evolving consumer demands for more environmentally friendly production systems. Just a few examples of these are, for instance, the directive from the European Council, where this was established in 2009. I think there has been some modifications recently, where the legislation really compels members or member states to really implement more measures that promote low pesticide input pest management, and give more priority to non-chemical methods. Similarly, United Nations members adopted this Sustainable Development Goals, where really the focus for our project is on Sustainable Development Goal number 12, which aims to ensure sustainable consumption and production. And in that SDG, one of the objectives is really to reduce the use of synthetic pesticides by 50% by around 2030. On the African continent itself, if we look at the African Union Agenda 2063, there's a call to develop strategies with the aim to grow the African blue and green economies. And again, the use of less chemical inputs speaks to this call. And so how do we find these alternatives to chemical inputs, or how do we contribute towards the reduction of their use? This can be done by tapping into naturally occurring biological alternatives. So if we look at a vineyard ecosystem, we know that the vineyard environment is home to a diverse community of microorganisms. Some of them contribute to grapevine health, and some may affect grape quality and wine fermentation. And within this ecosystem, therefore, you find these fungal pathogens such as Botrytis Cinerea, but they exist within this system together with their natural antagonists, which can be other filamentous fungi, yeast, and bacteria. And these natural antagonists have the ability to suppress the growth of these pathogens, which then speaks to an approach of biological control, which is the use of any living organism to combat a specific plant pathogen. And this is at the core of our project. And so biological control agents, or the natural antagonists, apply different modes of action to suppress the growth of these pathogens. This can be through direct mechanisms. So for instance, the production of antifungal compounds, so secondary metabolites that can inhibit the growth of the fungal hyphae, or the volatile organic compounds that can inhibit spore germination, cell wall degrading enzymes that can break down the fungal cell wall, or it can be through competition for nutrients. Some of these natural antagonists also act indirectly by inducing the host's natural resistance mechanisms, the plant host in this case. So biological control agents are not only desirable due to their antagonistic activity. Beyond that, they need to have other properties that allow them to be desirable candidates for application in grape and wine production. For instance, it's important that they can thrive in various environmental conditions, that they have different modes of action that are induced either simultaneously or in tandem, and also they should not produce compounds that are toxic or harmful to human health. And most importantly, because the goal is not always to eliminate chemical fungicides, but maybe to reduce them, it's important that biological control agents can exist or they are compatible with existing chemical fungicides. And of course, we most preferably would like to have biological control agents that do not have a negative impact on the final product, which is wine. And so looking at all these, we devised this project, as Iliana indicated, with two aims in mind. So firstly, to explore the biocontrol potential of selected microorganisms against Botrytis Cinerea infection of grapes in pre and post-harvest period, but also to investigate the possible effect of the presence of these BCAs in wine fermentation and on their impact on wine quality. And in order to increase the chance of finding desirable candidates, we looked not only at vineyards that use different farming practices, but also we looked at isolation of these microorganisms from different grape cultivars, from different grapevine tissues, and of course, from the Southern Hemisphere in South Africa and also from Italy, and therefore increasing the chance of having biological agents that have that quality of being adaptable to different environmental conditions. Our objectives in this case were very simple and direct. Firstly, not just the isolation of these microorganisms, but evaluating the modes of action in order to understand how these biological control agents inhibit the growth of Botrytis Cinerea. Look at the effect of the biofungicide, looking at how effective the biofungicides are by assessing their effectiveness in cell cultures, so in grapevine tissue cultures, and then also to look at their interaction between the biocontrol agents and desirable yeasts that are used in wine fermentation, and also potentially other wine spoilage yeasts. And also then to look at conducting wine production from grapes that have been treated with the biological control agents. So our project was divided into two work packages. So the first work package focused on looking at exploring the potential of the biocontrol agents. And in that we had three tasks. First, to isolate and characterize the potential biocontrol agents, to determine their mode of action or modes of action, and then to evaluate their effect in vitro plant experiments. Then secondly, the second work package looked at the influence on the wine fermentation process. Firstly, screening the selected isolates for desirable oenological characteristics, followed by evaluating their fermentation as single cultures, but also in multi-species consortia with Saccharomyces cerevisiae and potential wine spoilage yeast. I'm highlighting this task because this is an ongoing task that started at the beginning of this year, and the student in charge of this will share some of the results that emanate from task 2.1, and then task 2.2 will be continuing into next year. And then finally, looking at the production of wines as indicated with treated grapes. And so overall, the people involved will now share the results of the activities that they undertook. But I just wanted to highlight that this project helped us to build a solid research ecosystem that comprised junior students that were BSc and BSc honors students and interns that were actively involved in the collection of the data. And then master's students, again, also actively involved in data collection. Senior students that were PhD students that led quite a number of the research initiatives, as well as early career scientists, which are postdoctoral fellows that were not just involved in leading some of the research initiatives, but also mentoring and guiding junior students. So overall, looking at the different tasks, you can see that we had about 8 junior students that were trained, 2 MSc students situated at Stellenbosch University and UWC, PhD student at University of Milan who was also involved in the student exchange or staff exchange visiting Stellenbosch, and also other PhD students that did parts or collaborated in parts of the project. And of course, about 3 postdoctoral fellows at the different institutions were involved. Okay, so finally, I would like to thank all the participating partners and funders, and the individual students will share the actual results from these work packages. So, thank you.
Thank you, Evodia, for your presentation. Indeed, and thank you for the ecosystem under our collaborators and our, research groups. That is, of course, an invaluable legacy that goes well beyond, the scientific results obtained in this project. So we'll now move on the contribution of our partners, and you'll see that will be highlighted the interdisciplinarity richness of this project. Each presentation illustrates different aspects and elements of the research pipeline we established during the project. And this means from the microbial collection of the strains and attractive species, their characterization, wine fermentations, and of course the impact on wine quality. So let's begin with Arina Hitzeroth, who will present the identification of BCA candidates from South Africa fruit sources. So thanks, Arina. The floor is yours.
Good afternoon, everybody. I'm Arina Hitzeroth from the University of the Western Cape, and I was part of this bilateral project building. So I had 2 honor students. I had 2 students in part of the project. One was an MSc student and one was an honor student. So they set up looking for biocontrol organisms. So we mainly focus on epiphytic bacteria, looking for bacteria that's growing on the outside of the fruits, on the leaves as well as on the flowers of the fruits, and looking to see whether any of these bacteria have activity against Botrytis Cinerea. So some of these organisms, we searched for various fruits, and we had 2 locations that we sampled from. The one was in the Paarl area of the Western Cape, in the Kleine Drakenstein area, as well as an ARC farm in the Stellenbosch area. If you look at This is roughly the area that we sampled. The one is in the Paarl area and the one is in the Stellenbosch area. So we sampled fruit trees and some of the fruits, and we sampled some of the leaves as well. This was mainly the materials and methods that we followed. We isolated fruit. Sorry that there's only one fruit represented here, but we sampled various fruits that were found on these farms, including grapes, guavas, and some stone fruits. So these fruits were then sampled and isolated. We did in vivo assays, and then we isolated various strains that were positive, that showed positive activity against botrytis, and then we did some plate assays on these samples. So in total, we isolated about 100, the students isolated about 100 bacterial strains. Some of the strains, 6 were from some of the soil and rhizosphere area. Some we isolated 13 from fruit, 11 from leaves, and in total of these 100, only 16 of the strains had actually activity against botrytis when we did the in vivo assays. But when we did assay against various strains, only 7 of these isolates showed activity against botrytis. So these are just roughly the names of some of the species. Most of the species belong to the Bacillus group that showed activity against all of the botrytis strains that we tested against. Some were Pseudomonas and Priesta and Arwenia, which is normally associated with fruits as well as the leaf of fruit trees. This is just a recap of the in vivo assay that we did, where this was the Botrytis Cinerea control, and then some of the samples were then, the grape berries were sprayed with or treated with the bacterial isolates, and then it was challenged against botrytis. Then we did the in vitro assays to just see if we look at conidial suspensions, which strains had showed activity against the conidial suspensions as well as mycelial plugs. And in this, not all of the strains did really well. Only a couple of strains showed activity against various strains. So in the sample, it was only tested against one botrytis strain, and the sample R1B1, which was actually a bacillus strain, did the best of all the samples. In another, the MSc student did another assay where she looked at, and she tested it against 3 bacterial isolates, and they were some of the bacillus strains they tested really well and showed activity against all 3 bacillus strains. But most of the samples actually only showed activity against one, but only the bacillus strain showed activity against 3 of the botrytis strains isolated. We then further characterized these bacterial strains just to see if they have any enzymatic activity. And some of them did glucanase activity. And glucanase activity it indicates that it can break down the mycelial, the fungus cell walls. So the strain showed strong activity against glucanase activity. We also tested proteases, lipase activities, but these were just activities on plate screening. So we didn't do in-depth enzymatic analysis on these strains. But they did show strong activity against glucanase activity, but some only showed weak activity against chitinase, which forms part of the cell wall of the mycelia. Okay, so just wanted to wrap up, like these were the 3 students that were involved in this project. The first one was Malika Abraham. She graduated with an MSc earlier this year. And this is Axel Collins. He was the honor student in the group. And he graduated his honors degree as well. And this is my little lab group that we have started. Okay. So I just wanted to acknowledge for the NRA for the funding for this project. The Institute for Microbial Biotechnology and Metagenomics. I was part of the institute while doing this project, and I've since moved. And I just wanted to thank Ileana for allowing me to visit her last year during when I attended a conference in Europe. Okay, thank you.
Thank you, Arina. Okay, so not sure if we have time for questions. However, in case Francesca, you'll stop us. But it's now, I now give the floor to Dr. Heinrich du Plessis. He will show us the results from the microbial collection of potential BCAs on ARC farms.
Good afternoon, everybody, and thank you for the opportunity. My name is Heinrich du Plessis. I'm a researcher at the Agricultural Research Council based in Stellenbosch, and the title of my talk today is Isolation and Characterization of Yeast and Bacteria from ARC Farms. For you that's not familiar with the Agricultural Research Council, we're a science institute in South Africa, focusing and supporting and developing the agricultural sector. We've got different research institutes throughout South Africa. If you look at the picture on the left-hand side, we are basically based in the Western Cape in Stellenbosch, which is designated with a red circle. If you look at the picture, the right-hand side, there's just an enlarged picture to show basically where we are. We are based in Stellenbosch, the same, very close to the University of Stellenbosch. The circled areas also indicate the two farms that were sampled. We sampled Nitso Bay farm, which is the wine and grape farm that we have. And then we also have a Bindon farm, which does pome and stone fruit. We just wanted to see if we do get isolates from different fruit sources, not just grape, if we get a better biocontrol or similar biocontrol activity. So just shortly discuss the materials methods. So basically we sampled different fruit and grape sources. The fruit was washed and basically sterile, sampled and washed. For the bacteria, we basically played it out on plate count agar, nutrient agar, and Kingsby agar, which was supplemented with natamycin to prevent yeast growth. And for the yeast, we basically cultured on malt extract agar and Wallerstein nutrient agar, and supplemented with chloramphenicol to prevent bacterial growth. The isolates were basically plated out and subcultured and purified, and then cryogenically preserved in 20% glycerol. These isolates that we found were also then screened on grapes to see if they could inhibit Botrytis Cinerea FF1. All experiments were done in triplicate, and then just selected the isolates that showed the best inhibition or showed inhibition activity were then further screened in vitro assays, basically using the radial inhibition assay and the double petri dish measured for volatile organic compounds. The selective isolates basically was then also identified by amplifying the ITS region for the yeast and the 16S RNA gene region for the bacteria. So coming to the results part, basically, if you can look at the table, we basically sourced microorganisms from Muscat d'Alexandrie grapes, as well as leaves, Merlot grapes, Malbec grapes, and leaves. We had some peach fruit and also plum leaves and apples. In total, we isolated 63 microorganisms, of which we had 40 bacteria and 23 yeast strains. These strains were basically then used for the screening on table grapes to see if they could inhibit the Botrytis Cinerea growth. So basically, just to sum up, of the 63 isolates, only 10 of the bacteria isolates showed activity, and only 7 of the yeast showed activity, and so only the 17 isolates were further screened for growth inhibition in vitro trials. So the radial inhibition plate assay was basically performed to see if the microorganisms can prevent the growth and spore germination of Botrytis Cinerea. So basically, in short, we plated out on either PCA or PDA, media for the bacteria, and MEA and PDA for the yeast. So the control plate basically received 100 microliters of the specific microorganism, plated out at 1 million cells per mil, and the control plate basically only had the mold growing there. So inhibition was basically calculated by subtracting the growth of the treated plate from that of the control plate and expressed as a percentage. Come to the results. If you look at the left-hand picture, basically we see the growth of the bacteria on PCA. On the right-hand figure, we see the growth of the bacteria on PDA. It's interesting to note for the PCA, all the isolates showed 100% inhibition, whereas with the PDA, we saw more variability, and the bacteria didn't perform as well as in the case with the PDA, with the controls or the reference fungicide showing better activity. The best performing isolates in this case was D2 and MA. Moving on to the radial inhibition assay results for the yeast. The yeast was basically grown on MEA and PDA media. You can see there's less variability, and in general, the yeast performed well in inhibiting the growth against botrytis, with strains M1 and M3 performing the best, and they showed basically similar growth on PDE and MEA media, whereas other strains, like for instance B, performed better on PDA and not so well on MEA. Okay, then we followed up to see if the microorganisms can produce volatile organic compounds as a mode of action. So basically similar to the radial inhibition assay, the microorganisms were spread plated on the one plate, and the fungal spores or plug was put on the separate plate. The plates were basically put facing each other, then wrapped as shown in the figure in the left-hand side, and then left to grow. And then inhibition was basically calculated as the same as with the previous shown in the radial inhibition assay. So coming to the results for the bacteria, and in this experiment, basically we tested Botrytis spores. So if the bacteria could prevent spore germination as well, we also used the mycelial plug to see if the volatile compounds produced by the bacteria could inhibit mycelial growth. As you can see, most of the bacteria perform better in preventing spore germination, with less success with the mycelial plug, with the best growth in terms of spore germination being A13 with 78%. And for the mycelial plug inhibition, the best strain was J with about 44%. Coming to the volatile organic compounds produced by the yeast, as same as with the radial inhibition, the yeast showed less variability than the bacteria. And this experiment was the same as for the bacteria. We looked at preventing spore germination as well as the mycelial growth. The best treatments in this case was M5 and B, and they basically had similar activities for spore germination as well as for mycelial growth. There were exceptions where certain strains like D7 performed better with spore germination, but not as well as for the mycelial growth. But in most cases, the yeast were able to inhibit spore germination as well as mycelial plug growth. Coming to the identification results, on your left-hand table basically showing the results for the bacteria, and the right-hand table is the results for the yeast. As you can see, most of the isolates were obtained from grapes or grape leaves, and only 3 isolates were obtained from apple or peaches. Most of the bacteria isolates was either belong to the genus Pseudomonas or Klebsiella. There were also a few isolates that were not identified. On the yeast side, all the isolates basically were obtained from grapes or grape leaves. And the isolates basically that we identified was Pichia cluveri, Phyllobacidium orense, different Mexicovia species, as well as Oreobacidium pullulans. So just to conclude, only 27% of the isolates showed inhibition activity against Botrytis Cinerea FF1. The radial inhibition assay showed that media selection is important for bacteria screening and not so much for the yeast. In terms of volatile organic compound production, the bacteria were more effective in preventing spore germination, but the bacteria were not, or volatile compounds did not play a role in terms of mycelial inhibition. Yeast strains showed similar activity in terms of spore germination and mycelial inhibition in terms of volatile organic compound production. Most of the isolates that were sourced from grape or grapevine, indicating maybe that if the target organism is botrytis from a grape source, could be that it's just more effective against that specific organism. The yeast showed potential and should require further investigation. Then I just want to thank the people that actually did the work. So basically, I had 2 interns that rotated doing the work, and also 2 undergrad students that did most of the work, and they were supervised in the lab by my doctoral student, Zukezani Gemomo. So I just like to thank the team and also the different organization institutions for the resources and funding. And I also like to thank Prof. Zentini and also Prof. Setati for including me in this project. Thank you for listening. Thank you.
Thank you, Enrico. So now allow me to introduce you a couple of speakers. Hevelyn Maluleke and Christine Kruger will present together, results strictly connected to each other. Hevelyn will present the microbial diversity of South African vineyards and the role of BCA candidates in fermentation. And Christine will continue discussing the phenotypic traits of Mexicovia species relevant for winemaking.
Thank you, Eliana. Can everybody hear me?
Yes, perfect.
All right. So good afternoon, everyone. My name is Hevelyn Maluleke from Stellenbosch University, and my presentation is titled Microbial Diversity in South African Vineyards and the Role of Biocontrol Agents Candidates in Wine Fermentation. So building on what you've heard about isolating biocontrol candidates from various fruit sources, our focus in Stellenbosch University was specifically on the vineyard ecosystem. So we collected flowers, leaves, and grapes from vineyards that are managed differently, so biodynamic, organic, and conventional, to capture the full microbial diversity that is naturally present there. We isolated bacteria and yeast on different media. And what you see here is a summary of the number that we obtained from the different sources. So overall, we managed to retrieve a large number of bacteria and yeast isolates from the tissues that I mentioned, with yeast mainly coming from the grapes rather than the leaves or flowers, which makes sense since grapes provide sugar-rich environment, which is conducive for yeast growth. From our isolation, we were able to create a diverse pool of isolates that we screened for the antifungal activity. For screening purposes, we used Botrytis Cinerea as a model pathogen since it's easy to grow or to culture. And for the primary screening, we used disinfected grapes, which were gently wounded, and then we applied yeast as a preventative measure. after which, after 24 hours, we challenged them or introduced Botrytis Cinerea. We incubated the grapes at 25 degrees Celsius under high humidity. And in this case, our negative control was grapes which were inoculated with Botrytis only. And positive isolates were those that were able to inhibit Botrytis completely on grapes. So overall, we were able to identify 28 promising yeast isolates, plus 12 yeast-like fungus, which is identified as Oreobasidium pilalens, that we did not select for further screening as they are commercial products that already exist. And then on our bacterial isolates, we were able to identify 39 positive isolates against Botrytis Cinerea. So all the positive isolates that showed activity on grapes were identified using DNA-based methods, and identification relieved several bacterial isolates, including Pantoia agglomerans and Pseudomonas species, whereas when we look at the yeast showed more diversity with different Mechnicovia species, such as Mechnicovia hawaiiana, Mechnicovia pulcherrima, Mechnicovia ricaofi. We also isolated or identified numerous Hanseníaspora species, including Hanseníaspora pseudogilomundi and Hanseníaspora ovarum, among others. So all the identified candidates were considered for secondary screening, where we used the dual culture assay. For this, we placed our yeast or bacteria opposite Botrytis Cinerea, as demonstrated on this plate or image here, on malt extract. And for this assay, we tested all our positive isolates against 3 different strains of Botrytis, with one, which is FF1, confirmed as the hypervirulent strain. Also, this is the strain of Botrytis that we continued with for the entire characterization. From our secondary screening, bacterial isolates showed median inhibition of about 40 to 50% on average, whereas yeast performed even better, showing 44 to 55% inhibition. We also observed from this that some isolates were able to inhibit all the strains of Botrytis, suggesting they are broad spectrum biocontrol agents or candidates. So now, to understand how those yeast operate or inhibit the pathogen, we investigated volatile organic compounds that they produced, and for this we used the double petri dish assay, which we conducted as demonstrated here. So simply, we placed the yeast or the bacteria opposite Botrytis Cinerea on the agar plates, and then we sealed the plates to avoid the volatile organic compounds escape and also contamination. We incubated this for 5 days, after which we measured the mycelial radial growth. So overall, VOCs showed modest inhibition with inhibition percentage of around 24%, which was observed to be lower as compared to our direct assay that we conducted on dual culture. And this has been observed in other studies where lower inhibition is seen in such setup. Also, there are volatile organic compounds that have been proven to have antifungal activity, such as 2-phenyl ethanol, isoamyl, isobutanol, among other VOCs. Another mechanism that we looked at was the production of enzymes, so the chitinase and glucanases, as Botrytis cell wall contain chitin and beta-1,3 as major components on their cell wall. So we screened for those 2 enzymes as demonstrated on this image here. where we grew the yeast or the bacteria in the presence of botrytis, and the media is supplemented with either colloidal chitin for chitinase production or with laminarin for glucanase production. So overall, enzyme activity was widespread across the bacteria and yeast, with some different levels of observation. For example, all the Pantoia agglomerants were able to produce chitinase. However, not all of them were able to produce glucanases. We also looked at biofilm formation, which are communities that are able to attach to surfaces or to surface matrix. So this strong biofilm would indicate better colonization or longer protection on grape surfaces. So we did this assay using the crystal violet, which is a standardized procedure for measuring biofilms. We cultured our isolates in a liquid media, and then after incubation, we washed the cells that are not attached to the surface, and then used crystal violet to stain those that are attached, and then we measured the absorbance. So overall, we saw differences with bacteria showing much stronger biofilms formation as compared to yeast. On bacteria, we saw Pantoia and Klebsiella and Bacillus as well, producing the highest biofilms as compared to other bacterial species. So for practical application, we tested compatibility. We tested for compatibility where we used common fungicides, which in our case was the copper sulfate and switch fungicides. We tested this using YPD agar, which was supplemented with different concentration of switch or of copper, and then we inoculated our yeast or bacteria and then allowed them to grow. So the presence of a colony would mean that the yeast or the bacteria is able to tolerate that chemical that we are testing for. So overall, Pantoia agglomerants here tolerated both treatments well, whereas when we look at our yeast, all Hansennia spora species were able to tolerate all the treatments, so low and high concentrations of switch and copper. And then with Meshnicovia species, we saw that they were sensitive to switch, however, they could tolerate copper. So based on this characterization, we selected our strains that showed different modes of action against botrytis or against the pathogen. So this is the production of enzyme VOCs and biofilm formation plus fungicides tolerance. And then our selection included the South African isolates and also Italian strains that were considered for fermentation trials. So the selection criteria included origin, uniqueness, metabolite potential, and also whether they are registered as commercial products or not. And briefly, in South Africa, we selected Meshnicovia recaufi and Meshnicovia hawaiiana. And then from Italy, we selected Oreobasidium pilalens and Bacillus belezensis. So the selected biocontrol for fermentation trials. Now, for the selected biocontrol agents, we prepared them in liquid form at standardized concentrations, after which we treated our grapes that were freshly harvested. And then we divided the treated grapes into two groups. So one was left to dry for fermentation from dried treated grapes, and the other was hand crushed immediately to conduct fermentation from fresh treated grapes. Our fermentation was carried at 20 degrees, and we measured carbon dioxide daily to track fermentation progresses. We also took samples at the initial fermentation, so that day 0 until day 3, to plate out and see the microbial diversity that is there. We also took samples for GCMS, of which Daniela will present the data. So overall, we did see some differences whereby our fresh grapes were able to ferment a bit faster or shorter as compared to our dried grapes right here. And we also saw that Bacillus valezensis and Oreobasidium pilalens were top performance as compared to the 2 Meshnicovia species. And when we looked at the ethanol levels that we measured at the end of fermentation, we saw that the dried grapes showed a bit lower levels as compared to that of fresh fermented or fresh grapes. So to summarize, we identified promising agents that were effective against Botrytis Cinerea with different modes of action and compatible with fungicides. And then these candidates were also tested in fermentation trials. And I will now hand over to Christine to share what she did as she continued to characterize our Meshnicovia species.
Thank you very much, Eve. As Eve mentioned, I'm Christine Kruger, also from Stellenbosch University. And the title of the work that I will be presenting today is Phenotypic Characterization of Meshnicovia species and strains for application in winemaking. Now, this then brings us to the problem statement at hand. So while Empelkorima is well studied, the physiological and biochemical characteristics of newly isolated Meshnicovia species, particularly those linked to their fermentative abilities, remain understudied. So this is just a broader overview of my entire project, but for today, I will only be focusing on objective 1, which is to screen strains for enzymes of oenological relevance, SO2, and ethanol tolerance. So before every screening method, the yeast was first cultured on WL or YPD agar. They were washed with saline, and all cultures were diluted to get the same OD before plating to ensure uniformity when comparing different strains. So for characterizing the Meshnicovia species, I started with enzyme screening. I tested for protease activity on media prepared with skim milk, pectinase activity on media prepared with polygalacturonic acid, beta-lyase on media prepared with S-methyl-L-cysteine, which is an amino acid, then beta-glucosidase on media prepared with arbutin, and then HAS production on Biggie agar. For SO2 tolerance, wine B media was prepared, supplemented with glucose and varying concentrations of SO2. So I tested for 10, 15, 20, and 30 milligrams per liter. The cultures were inoculated at an OD of 0.3, and it was then incubated, and OD measurements were taken regularly to monitor the growth. So for ethanol tolerance, YPD plates were prepared, which was supplemented with different concentrations of absolute ethanol. I tested 5, 7, and 10%. So the yeast cultures, a serodilution were made from 10 to the power of 0 all the way down to 10 to the power of -4, and they were then spot plated onto the YPD plate supplemented with absolute ethanol. And for all these screening methods, the media, the pH of the media was adjusted to 3.5. So before I dive into the results, I just quickly want to show a visual representation of how negative and positive activity looks on these different plates that I screened the different enzymes for. So as you can see, for positive activity, in most cases, it is visualized with some sort of halo around the colony, except for beta lyase, which is only when the colony is present. And now for H2S production, this is the rating system that I utilized. You can see it is based on the color intensity of the colony streaked out onto the Biggie agar. Now for my actual results. For beta-glucosidase, only M. caryopsile and one M. bicus strain had positive results. Beta lyase, all the isolates had positive results except for the 2 M. hawaiiana strains. So for protease activity, I also utilized a rating system. This was based on the size of the halo around the colony. And as you can see, it's all in varying levels, except for 1 M. pulcherima strain and the 2 M. hawaiiana strains that did not produce any protease. Pectinase activity, there was none in any of the isolates I tested for. And H2S production is also all in varying levels, except for the 1 M. hawaiiana strain that had no H2S production. So moving on to ethanol tolerance, this is the rating system I utilized for scoring the ethanol tolerance. This is based on the ability of the yeast to grow at the different dilutions I made with the yeast. And the results that I obtained was at 5%, you can see that most of the isolates were still able to grow. At 7%, you can see they still struggle a bit more, but they are still growing. And then at 10%, only M. pulcherima, M. caryopsile, and 1 M. bicus pedata strain was able to grow. So the results of the SO2 tolerance, as I've mentioned before, I tested SO2 concentrations of 30, 20, 15, and 10 milligrams per liter. However, the Meschenkovia species was only able to tolerate 10 milligrams per liter, and there was absolutely no growth at all at 15 milligrams per liter. This was tested next to SRV713, which was my control, and at 15 milligrams per liter, SRV713 immediately went into exponential phase, while Meschenkovia species had a significant lag phase before it went into the exponential phase. So why did we test for these specific enzymes, and what is their relevance in oenology? So beta-glucosidase promotes the release of free terpenes, which leads to enhanced aroma complexity. Protease helps to prevent proteinase by degrading chitinases, and they are also known to free up nitrogen sources in the form of amino acids and peptides, which can then be utilized by Escherichia. And then beta lyase contributes to volatile flower release, which is responsible for fruity aromas in the wine. So when comparing SO2 and ethanol tolerance to literature, these strains have low SO2 tolerance and moderate ethanol tolerance. So when we look at the results that I've obtained so far, including the ethanol and SO2 tolerance, as well as the enzyme screenings, there are strains that hold potential for application in winemaking. However, it is yet to be determined whether these enzymes will be expressed under winemaking conditions. So just future work that is left for me next year is monoculture fermentations in synthetic grape juice medium, mixed culture with Escherichia in synthetic grape juice medium, and then also mixed culture with Escherichia in grape must. I'm also currently busy with testing for volatile phenol production, also in synthetic grape juice medium. So lastly, I just want to thank Reineke Wines, De Toren, Welgevallen Experimental Farm, SAF, and then also everybody else that was involved in these projects. Thank you very much.
Thank you, Hevelyn and Christine, for your interesting presentations. Now I invite Angela Conti, who will guide us through the microbial biodiversity found in Italian grapevines. Thank you, Angela.
Thank you, Nana. Thank you very much, everybody. I would like to be clear about my topic, which is obviously exploring microbial diversity in Italian grapevines. But I would like also to show the complexity of the biodiversity analysis, the technique that we use, just to show that it's important to know the issues to discover new and important microbial resources for BCA and sustainable viticulture application. So this is just a brief overview of my presentation. And I would like to go directly with just an analogy to understand why am I talking about the issues regarding the analysis, the exploration of the biodiversity. So exploring biodiversity is more or less like finding the responsible of a crime scene of a murder. We can have direct methods. If we are lucky enough, then a camera registered the person who was responsible for that. And we can also have indirect methods, which deals with a lot of signal. And if we are lucky enough, then we identify the person who is the murderer with the fingerprint, but there's an issue regarding the fingerprint, that is finding the right one among the lots of signals that we have. So this is more or less the same problem that we have while studying biodiversity. We have mainly two techniques to use: culture-dependent analysis, direct methods where we try to cultivate microorganisms directly on a plates. And it's good because it's direct. It's more or less like looking, like seeing the murder of a crime scene. But we have a limitation, that is the percentage of cultivable microorganism is less than 1%. And this is due to dormancy, inadequately replicated growth condition, or other physiological and environmental barriers. Again, we have also indirect methods, which are culture-independent analysis that rely mostly on sequencing techniques. And they are highly sensitive, so it's good. But as fingerprints for crime scene, we can also deal with a lot of signals with inadequate discriminations due to PCR errors, sequencing errors, or database incompleteness. And in this presentation, I would like to show how these two different methods were employed. Sorry? Okay. The two different methods were employed in the study of the biodiversity, the Italian biodiversity. And first, in Milan, they sampled two different species of vitis, leaf, berries, and flowers. And the total DNA was extracted, ITS amplification and 16X amplification was carried out, and then we sequenced the amplicons with mini-ion sequencing technique, which is good because we can reach longest fragments with this tool, with this instrument. What we saw is that there was no significant difference both among beta diversity, which is the microbial composition, among the conduction, the different management practices. And there was also no significant difference considering the community structure between the grapes. So there was no significant difference among neither the species nor the management practice. And we tried to look if there was a core microbiome, which is in this graph, we can see the microorganisms that were found in at least the 30% of the samples, with a relative abundance of at least 1%. And as you can see here, there were a lot of bacteria, both bacteria and fungi, that can be considered the core microbiome. And it's important to understand that because they could be a good candidates for BCA's analysis, as we will explore later in the following presentations. And with this, I would like to show also the difference between cultural and culture-dependent and cultural-independent analysis that we explored with this work. You can see that bubbles here are lots more, bubbles in this, in the graph here are a lot considering the culture-independent techniques compared to the cultural dependence, so the analysis of the microorganism that grows in the media. For ITS, and similarly, it's very similar, the same result we obtained with the 16S. This could be a problem of growth condition, as I said before. This could be a problem of speed of growth, considering the cells, and also a problem of abundances of cells within a sample. But the most important thing to look at in these two graphs is the difference between the results, the biodiversity that we obtain considering cultural independent, these two rows, compared to cultural dependent analysis. Just one fungus and one bacteria was found with both techniques. If we switch to the epiphytes community, so the community that's not inside the leaves but outside the leaves, we can add another layer of complexity. Here we analyze and we sequence also the epiphytic communities. We cannot see much differences here. The most important things to look at is that considering all the biodiversity that we can study, that we can measure, we see through cultural independent technique, sequencing technique, that there are species of, in this case, yeast that are reasonable microbiome of the viticulture ecosystem. And also we can find noise, so microorganism species that could not be there, but could be just a problem of sequencing. And the issues is due to, as I said before, the database that we use that can contain sequences that are not well, that are not good reference. It could be errors of PCR or sequencing, and it could be error of computational errors of matching. So what I would like to show is that either culture-based technique or sequencing-based technique have advantages and problems, but only a combined and complementary approach yields the most reliable and useful data. So it's important to combine the two techniques. That is what I would like to show with you. And a last thing to say is that considering the cultural independent technique, that they are very important to use in this case, the community, the technology can help us to go further with this analysis using not only ITS, but long-read DNA metabarcoding, which is a very interesting technique considering these studies and the importance of knowing the biodiversity that is in a site. So thank you very much.
Thank you, Angela, for your presentation. I think milestone in this search for biodiversity that is a hot topic nowadays. Is now the turn of Alessandra Di Canito. She will present the selection of potential BCAs and their interactions with grapevine and the pathogen, in this case, Botrytis Cinerea. Please, Alessandra.
Okay, thank you so much. Can you see my screen?
Yes.
Okay, thank you. Good afternoon to everyone. My presentation will be focused on the characterization and the study of the interaction of the potential BCAs with the grapevines and Botrytis Cinerea. In the context of the biocontrol, both yeast and bacteria isolated from the grapevine phyllosphere and endosphere emerge as highly suitable candidates. Epiphytes are microorganisms that live on the plant surface, while the endophytes reside inside plant tissues, either inter and intracellularly, without causing visible symptoms.
Their composition is shaped by several selective pressures, including geography, climate, pesticide use, and also soil management. Regarding the, the epiphytic strains that were used in the study, they belong to the University of Milan collection, and they were isolated between 2008 and 2016, mainly in Georgia, but also in Azerbaijan, Spain, and Italy. These yeasts were obtained from grapes, must, and wines deriving from both Vitis vinifera and Vitis sylvestris species, giving a total of 38 strains. For the endophytic collection, we isolated 148 strains, comprising 42 fungi and 106 bacteria that were obtained from both wild and domesticated grapevines from different regions and also different farming systems. They originated from different plant organs, including shoots, leaves, and berries. To characterize these potential BCAs, we firstly conducted in vivo experiments on table grapes. Each berry received a 5 millimeters wound, and after that they were submerged in the BCA cell suspension, and after 24 hours they were inoculated into the wound with Botrytis Cinerea. The symptoms were assessed after 2 weeks of incubation, and while around 34% of the inoculated berries showed sporulation, only 12% remained symptom-free. And notably, 3 epiphytes, Saccharomyces cerevisiae, Anseniaspora ovarum, and Gluconobacter bacterium, showed to be the best in this kind of experiments, the best potential BCAs, because they completely inhibited Botrytis cinereas in grape berries. Then we performed also in vivo assays on grapevine leaves. Leaf discs were obtained after sterilization, and then they were immersed in the BCA cell suspension and later challenged with a small 3 days old fungal plug. And after 6 days of incubation, we observed that Bacillus valenciensis showed the strongest inhibition of necrosis and mycelial growth, followed by the epiphyte Saccharomyces cerevisiae and the endophyte Aerobasidium pullulans. Subsequently, we performed also in vitro tests that were carried out on 3 epiphytes and 2 selected endophytes using dual culture and double Petri dish assays. Dual culture assays allowed us to assess competition for space and nutrients, and we observed that among the epiphytes, especially Gluconobacter showed strong mycelial inhibition, while among the endophytes, Aerobasidium pullulans and Bacillus valenciensis reached the inhibition levels up to 68%. While in the double Petri dish assays were used to evaluate VOC-mediated volatile organic compound-mediated inhibition. And the results revealed that the Aerobasidium pullulans achieved 52% inhibition, while the epiphytic community that we tested exceeded the 50%. But Gluconobacter bacterium reached the 62.2%. So to better understand this mechanism mediated by the volatile organic compounds, we evaluating the production of these compounds during the interaction between the BCA, the potential BCA and Botrytis Cinerea, using SPME GCMS method in a dual vial, medium vial for both the mold and the BCAs. Both obviously comparing the profiles of each organism alone and also in co-culture. Both epiphytic and endophytic yeast produce a broad and diverse spectrum range of antifungal VOX, including alcohols, ester, organic acids, aldehydes, and in some cases also terpenes. And the key metabolites were, for example, 2-phenylethanol, ethyl acetate, and medium chain ketones that could significantly impair fungal growth, germination, and also sporulation. Moreover, the pathogen-derived VOX were suppressed during the interaction, and the strains were grouped into 3 distinct metabolic clusters, reflecting the different interaction strategies or mechanisms. So, lastly, to demonstrate whether BCAs only inhibit pathogens or also influence plant immunity, the host immune responses were evaluated in Vitis sylvestris cell cultures, monitoring the expression of 2 key genes of the stilbene pathway. The STS1 that encodes for stilbene synthase, that is the enzyme responsible for resveratrol biosynthesis, and the ABCG44 that encodes for an ABC transporter involved in secretion and mobilization of phenylpropanoid-derived defense compounds. We monitored this expression after the inoculation of 2 different strains of Botrytis Cinerea, comparing the results when the mold was alone and combined with the Bacillus valenciensis, the selected Bacillus valenciensis, after 4 and 8 days of inoculation. The results were striking because in presence of the potential BCAs, STS1 expression was increased by 25 and 27-fold respectively after 4 and 8 days for the strain of Botrytis cinereas B0510. And for the UCA 992, the expression was slightly lower but still stronger. That was around 9 at 5 days and 5-fold at 8 days. Considering the transporter gene, we verified that the increase of the expression was modest, but it remained upregulated for about around 7-fold for the strain B0510 and 4-fold for the strain UCA 992. These findings clearly indicated that the selected potential BCAs act as a potent elicitor, enhancing both resveratrol biosynthesis and also its transport, thereby activating a sustained defense response in the phenylpropanoid pathway. All these results are part of the PhD work of Simona Pizzi, that are partially published, and also for a part we partially just submitted. And to conclude, these results show that altogether, epiphytic and endophytic microorganisms display strong potential as effective and sustainable BCAAs, and their combined abilities to inhibit botrytis through VOC production and also to prime plant immune defenses position them as promising alternatives to chemical fungicides. So finally, I would like to thank all the research groups that were involved in this project, and lastly, all of you for your kind attention.
Thank you, Alessandra, for your talk. We are approaching the conclusion now, but before to end, allow me to introduce you Dr. Daniela Fracassetti. She will share with us the results on wine grape quality and with grapes treated with the selected BCAAs. During her presentation. Please, Daniela, the floor is yours.
So thank you very much, Ileana. Good afternoon to everybody. So right now it's the time to have, after this very deep overview about the BCAAs, is the time to produce wine from grape treated from with BCAAs. So in particular, with these regards, the experimental design cover the two different harvests. In particular, in 2024, as Hevelyn presented, we carried out a trial considering Nebbiolo grape, both fresh and withered, while in harvest 2025, we consider Nebbiolo and Grechetto grapes. Differences between the two harvests was also related to the use of BCAAs, being 4 in harvest 2025 and 6 in harvest 2025. So I will focus my attention on the major results we got from the winemaking trials we carried out in harvest 2025. In particular, when we got the grape, the bunches was submerged into the BCAA suspension, and then they were left under the fume hood for drying. The must were obtained by hand pressing the bunches, and the only adjustment that was carried out was related to the ammonium in order to reach a content of readily assimilable nitrogen of 200 milligrams per liter. In all the cases, at least triplicate fermentations were carried out by using an active dry yeast for performing the alcoholic fermentations. Of course, as control, an aliquot of grape bunches were not treated with the selected BCAAs. In order to properly consider the the enological traits of the experimental wine we produced, we consider the general chemical parameters with regards of titrable acidity, pH, residual sugar, ethanol, and the organic acid profile. As well as we carried out a wine tasting considering different descriptors from the olfactory, taste, tactile, aftertaste point of view, as well as the pleasantness of the resulting wine has been taken into account. Let's see what we obtain. With regards to the Nebbiolo wines, the fermentation were completed in all the cases, independently from the different BCAAs used for the grape treatment. However, when we consider the fermentative vigor, some differences was evidenced, in particular with Anseniaspora ovarum, the highest fermentative vigor was found, while in case of Mechnicovia, Reucaufi, as well as the Pichia, the fermentative vigor was the lowest. However, in all the conditions, the fermentations, so the content of sugar, was lower than 0.5 grams per liter at the end. Then we consider in particular the content of ethanol. Ethanol, only little differences were found, and even if these differences were not significant, we found slightly higher content of ethanol in the presence of Aerobasidium pollulans, Anseniaspora ovarum, and Mechicovia hawaiiana. On the contrary, with regards of glycerol, we found higher concentrations still in the presence of Aurobasidium pullulans, as well as in case of Anseniaspora ovarum. Moreover, we also consider acetaldehyde as the precursors of ethanol, and we did not find any significant difference on the acetaldehyde released. Considering the acidic profile, actually was interesting to see that in case of Mechnicovia raucaufi, the content of malic acid was the lowest, as well as the lowest was the titrable acidity. Actually, looking for this strain for the alcoholic fermentation, only a recent research was published in which Mechnicovia was used for the production of cider, and in that case a decrease of the titrable acidity of malic acid was not observed. However, this characteristic can be also a strain-dependent characteristic. It should be evidenced that harvest 2025 was particularly strong in terms of acidity for Nebbiolo, as we got a relevant concentration of malic acid as well as of titrable acidity. The other strain being able to decrease the content of malic acid was also Anseniaspora ovarum, and our results was also in accordance with the previous literature, in which Anseniaspora ovarum have been described as a yeast being able to consume malic acid still in a strain-dependent way. None of the conditions considered lead to an increase of acetic acid, in particular reaching the content of the highest concentration of acetic acid was 0.34 grams per liter, lower than the concentration being responsible for a possible defect. From the sensory point of view, some differences were evidenced, in particular for certain descriptors, olfactory descriptors, in particular fruity and intensity, and some aftertaste descriptors, in particular vegetal and persistence. With regards to this aspect, the vegetal note was higher in the presence of Mechicovia hawaiiana, while the persistence was more relevant in case of Anseniaspora ovarum and Bacillus belenzensis. However, when we consider the pleasantness, the wines obtained with Pichia seem to be the most preferred in comparison to the other conditions investigated. some differences were found when we analyzed the Grechetto wines. Even in this case, the fermentative vigor were found different among the different BCAs investigated, with the highest value in case of the untreated grape bunches. On the contrary, the lowest fermentative vigor were found in peak with the treatment with Pichia. However, similarly to what was described for Nebbiolo grape, the fermentation were completed in all the cases. With regards to the content of ethanol, the difference were little and not significant, with slightly higher concentration in the presence of Aerobasidium pullulans and Bacillus valenciensis. With regards with the glycerol, similarly to what was found in case of Nebbiolo, the highest content was detected in the presence of Aerobasidium pullulans. Still, similarly to the results found for Nebbiolo, no significant differences were found in terms of the acetaldehyde concentration. With regards to the acidic profile, in general, lower content of both malic acid and titrable acidity were revealed in case of Grechetto wines. And actually, we could exclude a possible malic acid consumption being converted to lactic acid because in all the fermentation trials, its content was lower than 0.1 grams per liter. However, differently to the Nebbiolo wines, we did not find any significant difference, neither in case of titrable acidity nor in case of malic acid, based on the different BCAs treatment. Again, even in case of acetic acid, the content released during the alcoholic fermentation won't be associated to a possible defect in the resulting wine. With regards to the sensory profile, we found a decrease of the olfactory note of fruitiness when the grapes were treated with BCAs. And this decrease was found even in case of the aftertaste. Moreover, we found an increase of the vegetable note in case of Pichia, and even for the Grechetto wines, the wine obtained with Pichia seemed to be the most pleasant one. So, from the study we carried out, we can assert that the BCAs could affect the fermentative vigor. However, even if some differences were found, all the fermentation were completed, indicating that the treatment with BCA could not lead to a stuck fermentation. The BCAs were suitable for the grape treatment since no relevant differences were found in terms of enological parameters. We could observe a slight impact on glycerol being higher in the presence of Aerobasidium pullulans, while no significant difference were found neither for acetic acid nor for acetaldehyde. However, From the sensory point of view, we did register some impact based on the treatment with BCA, in particular for the olfactory fruitiness note and the vegetable note aftertaste. Such differences were more evident in case of Mechnicovia hawaiiana was used for the treatment of grape. However, it should be mentioned that the suspension used for the grape treatment was very concentrated, and so what should be carried out in order to demonstrate and to further highlight the limited impact of BC8 on wine production should be the application of BCA and at vineyard level, and then the production of larger volume of wines. So I would like to thank all the BiSus4Wine consortium, as well as Alessio Altomare and Denis Salieri, who are collaborating to this project, the funding agency, as well as all of you for your attention.
Thank you, Daniela, for your presentation. Indeed, also thank you for, your highlight. We are still at the laboratory level with our tests and experiments. So we hope to go in the field directly for next season. And we will need another project. However, so we are finished this session. So now a warm welcome to Florian and Gianluigi. So Professor Bauer and Professor Cardinali will handle this last session for conclusion and discussion. So you can.
Thank you, Ileana.
You're welcome.
Florian, it's up to you for the slides, I guess.
I think Evodia will share from her computer. Evodia, can you share the slide?
Thanks, Evodia.
Yes, I'll do that. Sorry.
So Gianluigi, you go first, huh?
Sure, thanks. So the gentleman agreement between me and Professor Bauer is that I take the difficult part of viticulture and he will take the more pleasant part of enology. No, I'm joking, of course. We would like to share some ideas, some general ideas about which we compared our points of view. And I will start with the viticulture. With viticulture, the first concept I would like to give you, also from a practical point of view, is that if in the last 10, 15 years, choosing BCAs was a choice in terms of having more organic wines, less chemistry and whatever, now it is a necessity because of the declining number of pesticides allowed in agriculture, especially in Europe. So in other words, if in the past we could have a choice, now we don't have it. We don't have any more any type of compound, any type of defense for our wines, especially in Italy. And this is particularly dramatic for organic wine, where the copper usage has been reduced to below 3 kilograms per hectare per year, which is below the actual possibility of using it. So first news and take-home message is we have the necessity to develop BCAs. And BCA, according to any other means of production, should be tested for efficacy and innocuity. But in normal cases, efficacy, okay, is if it is sufficient in reducing pests and whatever. Innocuity was considered against the humans, which is normally the case because most of the organisms are not able to grow at 37 or are not able to grow on human cells or human bodies over 37 degrees. So this is the least difficult part. The problem is the innocuity versus the wine, which is most of what also Daniela and others presented this evening, which is a matter of study, as you've seen, and a matter of scale up, because what we see in the laboratory is not necessarily what we will see in the wineries. In the wineries, we have to conduct the same fermentation at a different scale, and something could change. But what I would like to concentrate upon a single moment is the innocuity versus the environment. Because the problem is that if we use organisms that are able to dwell in the environment after being sprayed, and to substitute the biodiversity, the microbial diversity that was effectively present before the spraying, we are able to produce a microbiological drift in the environment, which could have effect that we are not able to imagine or to predict. This means that at least in my and I think also Florian's view, I compared this concept also with some guidelines coming from the Ministry of Agriculture in Italy and from other places. Some care should be given to the fact to assess whether these organisms would remain and would, let's say, destroy or change significantly the existing biodiversity. Then the other point, which is important for research, we speak about BCAs, but the problem is whether they are competitor or antagonist. The preference should go for competitors for the space, which is somehow also competition for the nutrients, because the space is not significantly different from nutrients onto the fruit surfaces. But the advantage of competitors versus the antagonist is that they stay as long as there are sources of sugar. When the sugar is gone, they leave. Whereas the antagonist could remain and could affect more significantly the fermentation. And the last two remarks are for our young cooperators that did most, if not all, the work at the bench, is that the BCA, if allowed, and there is a long way for having them registered or allowed, not all legislation will be very easy to change, will need much more skill than chemicals asked for. And therefore, we need really prepared microbiologists, not only technologists, for dwelling with these things. And a final remark that is also a question that I leave to the audience, to everybody for meditation is, should we go for universal BCAs or should we develop techniques to pick up the right BCA in specific vineyards and use them in the same vineyards? Because in the second case, the change of biodiversity would not be significant or dramatic, at least it's something that was already there. In the first case, what we risk is exactly what happened in the last 60 years on the wineries. 60 years ago, you know, many wineries did produce very poor type of wine, and people introduced selected yeast strains, not thinking that those few, probably 10, 20 strains in the world would take out all the existing biodiversity, and the flatness in the diversity of wines is something that has been discussed over the last, I would say, 30 years. So we did not know, at least I would say our father in science did not know this effect 30, 40, 50 years ago when they introduced the starters massively in the wineries. But now we know that this effect can exist. And we should refrain from doing the same mistake because at least we know. And maybe picking up the diversity and selecting it efficiently from the same place where we want to spray it, it is a choice. And there are paradigms like this effective in soil microbiology. And I think that I give the floor to Florian because the rest is enology.
Yeah, thank you, Gianluigi. So I don't want to speak much about enology. because in enology, if you think about the process of winemaking, actually the whole winemaking process is designed to control the microbial ecosystem. Basically, all the interventions that people do are primarily undertaken to control the ecosystem, whether it's SO2 addition, inoculations, of course, are a direct intervention in microbial biodiversity. Nutrients are there to also favor certain organisms. Oxygen treatment, the same. Temperature control controls the ecosystem. So winemaking, from that perspective, you could think about it as a biocontrol enterprise. You try to control which organisms are conducting the fermentation. You try to control the speed at which they do it. So the whole winemaking process is, by definition, in this context, a biocontrol enterprise. That doesn't mean we don't need to improve on what we have, and we still need sustainable tools to control, particularly some unwanted species. Brettanomyces is always cited, so we can certainly make progress in that. The point I wanted primarily now to focus on is what are the major scientific and application challenges that we have. We have seen here in all of the presentations, we are pretty good in identifying individual organisms and identifying mode of actions. Although for all the presentation, you can always argue, did we really show that this specific mode of action is actually valid in the context of biocontrol? We don't know, actually. And that is a major, major hurdle that we really have to overcome. And this is, we actually do not, and I repeat, we do not understand the microbial ecosystems that we try to control. We have good data. Angela spoke about biodiversity. So we have excellent now data on what species are around, where they are, but we actually don't understand how the system works. And that makes it actually very hard then to predict, of course, what the impact of a specific BCA, biocontrol agent, will be. We actually do not know how it will impact in the vineyard. So Gianluigi already spoke about this aspect in terms of what does it do if you actually release a BCA. If you think about mammals, it's easier to imagine with mammals because we see them and we can control them easier. Think about Australia when they try to get rid of mice, so they released cats, and what happened? Well, the cats eliminated all the native birds because they were easier to catch than the mice. So the question is, when we release BCAs in high numbers, what will it do to the local ecosystem? We don't know. That's not to say we shouldn't try and we shouldn't use this, because we need to get rid of chemicals, and we know that chemical use is not sustainable, but there are certainly inherent risks, which at this stage we actually are unable to assess. We don't know, as simple as that. Think about introducing a mammal somewhere and you would face major, major issues, ethical issues. We microorganisms, we tend to think, well, we can just use them. Can we? We don't know. I want to raise this as a question. It's not to scare anybody, but these are serious things that we have to look at. And then the final point I want to make is indeed one that Gianluigi already made in his comments as well. And that is that I don't think that we will be able to come up with really some generic BCAs that will work in every single context and that we can just spray wherever. In most cases, these approaches will require local adaptations. We will need to adjust it to the local ecosystem, to the locally present microbiota, and adapt. So that comes also then back to the fact that it will require quite a high level of skills to use these tools in an efficient and responsible way. So I wanted to give a slightly more critical assessment to what we are doing. We have to really carefully consider all of those implications. Yeah, thanks. I don't know if there are any questions.
Thank you, Gianluigi and Florian, for your critical point of view. But I mean, it's always welcome to be critic enough. I must say that No, that's true. That's true. No, I think, I mean, we are probably on a new highway, driving a new car, but with a lot of traffic, and we don't know really how to manage everything. So I think now we can conclude our event for today. Evodia, are you here with us? You can show you in the screen.
Yeah, I'm still here.
Yeah, okay, okay. So I think, would you like to add anything to the discussion?
Not really, but I mean, I agree to some extent with Florian and with Gianluigi, especially when it comes to real life application of the biocontrol agents. We did a study last year with one of our master students with other biocontrol agents to really do field applications in the vineyard and to see you know, even whether you detect the organisms that you would have applied in the vineyard, in grape must and in a fermentation process. But of course, again, we applied these, for instance, 2 weeks before harvest, which is still not the way the biocontrol agents would be used. And but we could see that depending on the species that you have, some of them will really be abundant, and luckily with the organisms that we were working with, they are not very strong fermenters and therefore they don't perturb the fermentation ecosystem. But then you don't know long term how they would perturb the natural microbial population in the vineyard and on grapevine. And I think that's important to really look into and understand before a selected organism can be even submitted for potential registration as a commercial product. But I think we have to start somewhere. And as you said, we are on a highway driving a new car, but we will get there. We will get there. Certainly,
Yes.
all the points that were raised are valid and need to be considered.
Okay, also thank you for this point of view. That is indeed in the middle, and it's the fun part of the research, I mean, I think. So to conclude the event for today, again, thank you for for attending the meeting. And we would like to express our gratitude to the funding institutions, to the Ministry of Foreign Affairs and International Cooperation in Italy, and the National Research Foundation in South Africa, all partners, universities and research institutes, the laboratory teams, and the students involved in this project, and of course you for to be here today. So thank you for sharing this journey with us, and have a nice evening.